Hey everyone, and welcome to This Explainer. Today, we're diving into an explanatory analysis of something pretty spectacular. We're going to completely reframe how you look at biology. Now, we've all heard the cell called the basic unit of life, which, let's be honest, sounds a little dry, doesn't it? So instead, I want you to picture the cell as this bustling, super dynamic, microscopic metropolis. We're talking borders, a central government, industrial factories, even recycling centers, all humming along in perfect sync. Which brings us to the big question. What is the fundamental blueprint that actually makes up every living organism? And, this isn't just some abstract philosophical musing. This is the exact mystery that kicked off one of the absolute greatest scientific revolutions in human history. Decoding this blueprint literally means figuring out how life itself ticks, minute by minute. So let's take a really quick look at how we figured this out. The whole thing kicked off way back in 1665. Robert Hooke was peering through a self-made microscope at a piece of dead cork, and he spotted these tiny, empty little compartments. He called them cells. Then, fast forward to the 1830s, Schleiden and Schwann realized, wait a minute, this isn't just a plant thing. They formulated this cell theory, basically saying, hey, all living things are built out of these structures. And then, taking a massive leap to the 1960s, Lynn Margulis totally flipped biology on its head again. She proposed the endosymbiotic theory, which showed that parts of our own cellular city actually used to be free-floating ancient bacteria. Wild, right? Okay, here's our roadmap for today. Number 1, we'll look at basic cell types. Number 2, membrane and transport. Number 3, the control center, which is the nucleus. And finally, number 4, production and energy. Let's get right into it. All right, section 1, basic cell types, foundational blueprints. Just like human cities, think of the rigid grid of New York City versus the totally winding, chaotic streets of London. Cells are built on drastically different foundational blueprints, and it all depends on what their job is in nature. Let's compare our two major eukaryotic cities, plant and animal cells. Plant cells, they're basically fortresses. They've got this super rigid cell wall that gives them a fixed rectangular shape, and the whole city centers around this massive water tower called a large central vacuole. And when they need to divide, they actually build a wall right down the middle called a phragmoplast. Animal cells, though, are way more flexible. They've got an irregular shape, much smaller vacuoles, and these really unique structures called centrioles that step in to orchestrate cell division. It's a perfect example of architecture dictating function. But if we zoom way out to look at the entire tree of life, the divide gets even bigger. You've got prokaryotic cells, like bacteria, which are honestly more like sprawling open-plan villages. There's no nuclear membrane, so their circular chromosomes just float freely, and they move around using these whip-like tails called flagella, made from a simple protein called flagellin. On the flip side, eukaryotic cells are the massive, complex metropolises. They lock their linear chromosomes safely away inside a true nucleus, and they use way more complex tubulin structures to get around. And here is where it gets super interesting. There's this subtle but massive difference in their machinery. Ribosomes are basically the protein builders of the cell. Now, eukaryotic cells use a larger 80S ribosome, while prokaryotes use a smaller, slightly different 70S version. Why does this matter? Well, this tiny structural difference is literally how a lot of antibiotics work. They're designed to specifically target and shut down those 70S ribosomes in bacteria, completely ignoring and leaving our own 80S ribosomes completely unharmed. It's an incredibly precise molecular strike. Moving right along to section 2, membrane and transport. We're talking about the city borders. Look, no city survives without strict border control. You have to dictate exactly what raw materials get in and what waste gets out. This border is beautifully captured by something called the fluid mosaic model, which Singer and Nicholson introduced back in 1972. It's important to realize this isn't a solid brick wall. It's actually a dynamic, shifting sea of phospholipids. Floating around in this fluid sea are proteins that act as literal transport gates, along with carbohydrates, collectively known as the glycocalyx, that function as the city's recognition signboards. And weaving through all of this, keeping the whole thing together, is cholesterol. It acts as a stabilizer, so the border doesn't melt when it's hot or snap when it's freezing. So how does cargo actually cross this shifting mosaic? Well, it all comes down to energy. Passive transport is exactly what it sounds like. It requires zero cellular energy, no ATP. Stuff like oxygen just naturally flows down its concentration gradient, basically slipping right through the border. But active transport, that requires ATP. The cell has to spend energy to actively pump molecules against the gradient. A perfect example is the sodium-potassium pump in our nerve cells. It's constantly burning energy to throw sodium out and pull potassium in, just to keep the cell electrically charged. All right, Section 3, Control Center, the nucleus. This is the central government, the highly secured vault where the ultimate instructions for every single function in the city are stored and heavily protected. This nucleus is wrapped in a double-membraned nuclear envelope, and it is studded with these highly selective pores. The import-export rules here are incredibly strict. Things like messenger RNA and ribosomes are granted clearance to exit into the city, while vital enzymes are allowed in. And deep inside this vault is the nucleolus, specifically a region called the nucleolar organizing region, which is basically a massive factory churning out those ribosomes we talked about earlier. Now, most of our cells are mononucleate, meaning they just have one nucleus, but our hardworking skeletal muscle cells are actually multinucleate. They hold many nuclei just to keep up with their massive energy demands. So the crucial takeaway here is how this genetic data is stored. It's not just a messy pile of DNA shoved in a drawer. It's highly categorized. Euchromatin is loosely packed DNA. Think of these as the open, active files sitting on the desk that the cell is currently reading to build things. Heterochromatin, on the other hand, is tightly condensed and totally silent. These are the archived files, permanently packed away on boxes because this specific cell doesn't need those instructions for its particular day job. Which brings us to Section 4, Production and Energy, the industrial zone. This is where those blueprints are turned into physical reality. Materials are synthesized, and the immense power needed to keep the city running is constantly generated. Let's map out this biological assembly line. Step 1 is the endoplasmic reticulum, where the initial raw synthesis happens. From there, materials get boxed up into tiny transport vehicles and shipped over to Step 2, the Golgi Apparatus, for modification and packaging. And finally, Step 3 involves sorting all these finished products, a process that includes manufacturing lysosomes, which are the cell's highly potent waste management and recycling centers. Let's zoom in on that first step, the endoplasmic reticulum. It's actually split into two very distinct districts. The smooth ER looks like a branching network of tubes. It has no ribosomes, and it's totally dedicated to synthesizing lipids, detoxifying harmful chemicals, and storing calcium. Literally right next door is the rough ER, and it is completely studded with ribosomes. Because of all those ribosomes, its main, absolutely vital job is the mass manufacturing of proteins. Once those raw proteins and lipids are built, they are shipped straight to the Golgi complex. Think of the Golgi as the city's central post office. It receives these raw materials at its entry point, which we call the sys face. Once inside, it performs some really brilliant modifications. For example, glycosylation, which is basically slapping carbohydrate shipping tags onto proteins. Finally, it ships these finished, polished products, like cellular mucus or digestive enzymes, right out of its exit port, the transface. Now, some of the absolute most dangerous packages shipped by the Golgi are those lysosomes. Inside, they maintain a highly acidic pH of around 5.0. To put that into perspective, it's a severely harsh environment packed with about 40 different hydrolytic enzymes, all specifically designed to aggressively tear apart biological molecules. But because of this immense destructive power, lysosomes are the ultimate recyclers and protectors of the city. They handle phagocytosis, which is essentially eating and digesting foreign invaders like bacteria. They do autophagy, eating up old, broken-down cellular machinery like damaged mitochondria. They even act as suicide bags, triggering apoptosis, or programmed cell death, if the cell becomes a threat to the body. And this is critical. If a person is missing just one specific lysosomal enzyme, like hexosamididase, lipids will fatally accumulate in the brain. This causes incredibly severe conditions like Tay-Sachs disease. So yeah, these tiny acidic sacs are absolutely vital to your survival. Finally, we arrive at the powerhouse. Mitochondria are the furnaces of the cell, originating from ancient aerobic bacteria. This is that brilliant insight from Lynn Margulis in action. Mitochondria actually have their own circular DNA and their own 7DS ribosomes. They perfectly match bacterial profiles. And just because of how human fertilization works, the mitochondria keeping your cells alive right now were inherited entirely, 100% from your mother. Their structure is perfectly engineered for massive energy production. The inner membrane is folded into these structures called cristae, which massively increases the surface area. Embedded all along those folds are ATP synthase particles just spinning furiously to generate power for the entire city. And inside all of that, the matrix holds the mitochondrial DNA and the machinery needed to keep this localized power plant running completely on its own. So there you have it. We've mapped the borders, we've visited the government vault, toured the industrial factories, and stood right inside the power plant. Think about it. You have a bustling, incredibly complex metropolis operating inside you, multiplied by trillions every single second of every single day. How does that change your perspective on life? It really is an absolute engineering marvel, and it's happening inside you right this very moment. Thank you so much for joining me for this explainer. Keep wondering and keep learning.